The purpose of this research was to learn more about the chemistry that led to life on Earth. The research focused on prebiotic chemistry and its evolution that allowed the building blocks of life to form. Scientists analyzed the conditions that allow the formation of peptides from amino acids. An important method was condensation reaction, which created similar conditions to Earth billions of years ago. Proteinoids were formed through a higher temperature condensation reaction that allowed the specific peptide to form. For many years, the theory of proteinoids was valid, but it has been replaced by the RNA world theory as a new theory that gave rise to life. Condensation reaction at different temperatures led to deoxypeptides, which contain one or more ester bonds, and they are peptide chains formed from a reaction between amino acids and hydroxy acids. As the result of this experiment, 7 successful deoxypeptides were formed. This experiment connects to several biology modules and allows us to apply our knowledge in real-life experiments.
There are chemical similarities between peptides and deoxypeptides. To clarify, deoxypeptides are peptides with 3 amide groups and 1 ether bond group. In this experiment, a nomenclature system was developed to designate monomers and residues. Thus, a peptidomic workflow was developed for mapping the sequence diversity of deoxypeptide libraries generated from combinations of plausibly prebiotic monomers. Scientists simulated deoxypeptide sequences and compared them with possible permutations using homology diagrams. Scientists believed that the simulation would be an accurate representation of the conditions that supported prebiotic life. After utilizing this workflow, the scientists were able to map the deoxypeptide chemical space. These maps enabled comparisons of relative abundance, composition, and hydrophobicity of the 3 types of linear deoxypeptide systems examined. Overall, the results revealed highly complex mixtures with hundreds of detected species with different permutations, even after a few environmental cycles. One potential future study involving protopeptide chemical space to find different deoxypeptide sequences and permutations would be to research deviation from ion mobility trends. The resulting characteristics could potentially indicate the emergence of secondary structures in said sequences.
Next, looking at deoxypeptide sequence isomers, the experimenters wanted to create a steady workflow for sequencing the entire set of isomers. To do so, they took a small subset of the isomers to begin with, analyzed the length of this subset, and then replicated the process on a large scale so that they could find similar results for the entire set of data that they were working with. After getting their experimental results, they then compared them to the theoretical results that they had expected. The experimental protopeptide results revealed a high sequence diversity of plausible monomer combinations for the entire experimental data. This experimental result led to the idea of an early life model more accepted than previously found through theoretical data. The experimenters were surprised by these results that they found.
As for the implications of the research, the experiment looks into the origin of life. In the past, scientists have had a tough time agreeing on the pathway to life. Knowing the diversity of the prebiotic sequences allows for better overall knowledge towards origin possibilities.